Excelligent's real-time cell analysis enables you to quantitatively monitor the killing of cancer cells by immune cells under label-free conditions and over the course of multiple days. This system provides a complete time course of target cancer cell killing, allowing the simultaneous screening of a large number of constructs, effector cell types, or combination therapies at low effector to target ratios.
Begin by seeding 1.5 times 10 to the 7 HEK293 FT cells in DMEM culture medium in a 150 mm cell culture dish for an overnight incubation at 37 degrees Celsius and 5% carbon dioxide with humidity. The next morning, mix 2.5 milliliters of transfection dilution solution supplemented with 5 micrograms of lentiviral vector plasmid DNA and 22.5 micrograms of lentiviral packaging mix with 82.5 microliters of transfection reagent in 2.5 milliliters of transfection dilution solution. After a 15-minute incubation at room temperature, transfer the reaction dropwise into the dish of HEK293 FT cells and return the cells to the incubator for a second overnight incubation. The next morning, replace the supernatant with 19 milliliters of fresh DMEM culture medium and return the cells to the incubator overnight. Collect the supernatant into a single 50 milliliter conical tube for 4 degrees Celsius storage for the next 2 days. Before sedimenting the lentivirus by centrifugation on the second morning in order to remove any cells or debris from the solution. Transfer all but about 1 milliliter of the lentivirus containing supernatant to an ultra-clear centrifuge tube for ultracentrifugation. After ultracentrifugation,